Good afternoon, listeners. I am Dr. Sangeeta Pandey from Amity University, Uttar Pradesh, and we are going today for a very important session that is on biofertilizer. So, as we know that first we will talk about what is biofertilizer and why it is important. So, actually biofertilizers are biofertilizers are those substances which contain living organisms, and when we apply these living organisms in the seed or in the soil or in the plant, then what happened? They colonize the root around rhizosphere, that is around the root, and then they help in better growth and development of the plants. So, we can say that biofertilizers are very important because as we know that these days use of chemical fertilizers are increasing, and as the use of chemical fertilizers are increasing, we see that lot of contamination is there around the environment, and lot of contamination is there around the environment and in the soil. Now these use of pesticides, or you can say use of these chemical fertilizers, are discouraged, and now we want to use this biofertilizer kind of thing, so that we okay, sorry, actually slides were not moving. So now we can start with the proper thing. So actually why we are talking about biofertilizer these days? The biofertilizers, since the green revolution, we know the use of these chemical fertilizers have increased beyond a limit, and then what we have done that after that during green revolution we introduced the varieties, high yielding varieties of rice and wheat, and then we saw that these varieties were highly responsive to the use of fertilizers, and when we started over applying these fertilizers, our environment, our soil, our food, everything got contaminated. So these days biofertilizers are very much in talk, and we want to know about biofertilizers, and we want to promote the use of biofertilizers among the farmers. So we have seen that this overuse of chemical fertilizers and pesticides have not only degraded the land and water, but also they have led to generation of various kind of diseases. So we can see that these biofertilizers are very important, and these biofertilizers have ability to replace the use, not completely replace, but reduce the use of chemical fertilizers and pesticides. Also biofertilizers not only supply the nitrogen, phosphorus and potassium to the plants, but also they have capability to supply other kind of micronutrients like zinc, iron, and as we know that there are 17 essential nutrients or elements which are required by the plants. But what we do in the form of fertilizer, we apply only nitrogen, phosphorus and potassium. We don't take care of the other trace metals or other micronutrients which are required by the plants. But these biofertilizers, when we apply these biofertilizers, these are having capability not only to mobilize the use of this nitrogen, phosphorus and potassium, but also they have capability to use the make zinc and iron available to the plants. The microorganisms in biofertilizers restore the soil's natural nutrient cycle and build soil organic matter. Through the use of biofertilizers, healthy plants can be grown while they enhance the sustainability and the health of the soil. They are extremely advantageous in enriching the soil fertility and fulfilling plant nutrient requirements by supplying the organic nutrients to the plant. Biofertilizers do not contain any chemicals which are harmful for the living soil. So we can see that there are numerous advantages of the biofertilizers, and when we start applying these biofertilizers and when we promote these biofertilizers among the farmer, we can see the tremendous benefit. We can see tremendous benefit of this. Now what are the various kind of biofertilizers? So we can see that the biofertilizers are categorized majorly as nitrogen fixing biofertilizers, phosphorus solubilizing biofertilizers, then phosphorus mobilizing biofertilizers, potassium solubilizing biofertilizers, PGPR based biofertilizers, and organic matter decomposers. So First, we will talk about the nitrogen fixing biofertilizers. These are the most common one, and among these, I think all of you are aware with the Rhizobium. So, Rhizobium is the most common nitrogen fixing biofertilizer, and it forms symbiotic association with the root of the leguminous plant. Symbiotic, when we say symbiotic, it means symbiotic relation, that both benefit from each other. When this association is there in between plant roots and the Rhizobium, then both get benefited. What happens? Rhizobium provide nitrogen to the plant, and at the same time plant provides shelter and food to the Rhizobia. So, Rhizobia, on the basis of their growth pattern, they are further classified in 4 groups, that is fast growers, Rhizobium, then slow growers, that is Bradyrhizobium, stem nodulating Rhizobium, that is Azorhizobium, and the Cynorhizobium. Now, Rhizobium are, I would like to tell you that Rhizobium are very specific to the type of legumes they are grown. They are very specific to the different species of, we should say not only genus, but they are specific to the species of the your crop which you grow in your field. So, on the basis of that, this cross inoculation group of Rhizobium, they are further classified as, like, for clover groups we apply this Rhizobium trifoli that infects and nodulates the plants of genus Trifolium. Second one is alpha group, that is Rhizobium meliloti, that infects and nodulates the roots of this Medicago and Melilotus. The third one is bean group. So, Rhizobium fascioli infects and nodulates plants of the genus Fasciolus. Then lupine group, in Rhizobium lupine, they nodulates the lupine. Then P group, they Rhizobium leguminosarum, they infects and nodulate the pea, sweet pea, lentil and vetch. Similarly, cowpea group and soybean group, that is Bradyrhizobium japonicum, that is the slow growing Rhizobacteria, and they nodulate the glycine and the soybean. And similarly, cowpea group is also there, and they nodulate the cowpea, pigeon pea and groundnut, among few other leguminous species. So, other important group of nitrogen fixing bacteria is the Acetobacter. So, Acetobacter digotrophicus is an endophytic nitrogen fixing bacteria, and they are found inside the root, stems and leaves of the sugarcane. So, when we say that it is endophytic bacteria, it means they reside inside the plant tissues. So, majorly they are found in, sugarcane crop, and it has the potential to fix around 200 kg of nitrogen per hectare. It is abundantly present in xylem, sap of sugarcane, leaf, stem and other tissues, and they are capable of growth at pH 3. It has ability to fix nitrogen even in the presence of nitrogenous fertilizers, which makes them distinctly different from other digestropes whose nitrogenase enzyme they succumb to the low level of nitrogen. So, we can see that this Acetobacter is different from other nitrogen fixing bacteria because their nitrogenase remain active even in the presence of nitrogen. So, generally we see that when we in leguminous crop, we do not recommend to apply the nitrogenous fertilizer, because if nitrogen is there in the atmosphere or in the soil, then Rhizobia which is present in leguminous crop, they will not fix the nitrogen. But Acetobacter is an exception to this, because Acetobacter has the capability to fix the nitrogen even if nitrogen is present in the soil. Other important group is Ajospirillum. So, Ajospirillum is a free living microorganisms. They are associated with roots of cereals, millets and sugarcane. They fixes nitrogen with the help of nitrogenase enzyme, which needs microaerophilic condition. Microaerophilic condition why we require, because nitrogenase is an enzyme which is responsible for fixing of the nitrogen, and they are very sensitive to the presence of oxygen. That is why we try to maintain either anaerobic condition or microaerophilic condition inside the bacteria which fixes the nitrogen. Also, Ajospirillum has the ability to fix 20 to 40 kg nitrogen per hectare in association with the root. It also produces plant growth promoting hormone like auxin and gibberellin, which further helps in improving the plant growth. Other important group is Ajola. It is a fern which acts as a host for the BGA, blue-green algae. Ajola is a, like we can say, this is a fern which acts as a host for the blue-green algae, and this is host for cyanobacteria, we can say Anabaena ajoli, and nitrogen fixation is attributed to the Ajola actually carried out by the Anabaena. So, Anabaena ajola is a kind of association, symbiotic association between fern and cyanobacteria, and which is generally applied in the rice field because they require water for their proper growth and development. And it has been found that inoculation with Ajola in a paddy field can fix almost 25 to 40 kg of nitrogen per hectare per crop. So, Ajola contains actually other than this, Ajola has nitrogen, phosphorus and potassium. So, when we apply Ajola in the field itself, they helps in enriching the, not only they fix the nitrogen, but also they help in enriching the soil with various kind of nitrogen, phosphorus and potassium, and other than this they also enrich the soil with organic matter. It can be grown in other crop also, not only in rice, we can use this in other crops if there is availability of water in the field. Now, Agitobacter. So, Agitobacter is a very important group of nitrogen fixing bacteria, and we can say that this is free living nitrogen fixing bacteria because they are not associated with any crop, they do not form any symbiotic association with any crop. They are freely present in the soil around the rhizosphere, and they can be used in, so we can say that they can use in wide range of crops. So, Ajola is basically used in cereals, especially wheat, and Agitobacter is used in cereals, especially wheat, and it also synthesizes appreciable amount of growth promoting substances including vitamins, auxins and gibberellins. It also helps in augmenting the germination of the seeds in various crops, and it has ability to fix 20 to 40 kg nitrogen per hectare in the field. Now, the other important group of the this biofertilizer is phosphorus solubilizing bacteria. So, apart from nitrogen fixing bacteria, we can safely say that phosphorus solubilizing bacteria is one of the most important group of biofertilizers which are not very commonly used, but this is very important to increase the availability of the phosphorus in the soil. So, we can say that these are the beneficial bacteria, they are capable of solubilizing inorganic phosphorus from the insoluble compounds. Now, P-solubilization ability of the rhizosphere microorganisms is considered to be one of the most important trait that is associated with plant phosphate nutrition. Actually, what happens, whatever phosphatic fertilizer we apply in the field, whether it is single superphosphate or double superphosphate, whatever this phosphatic fertilizer we apply, they do not become available to the soil unless the pH of the soil is correct. So, phosphorus becomes available in the nutrient range only, neutral range only. So, when the soil is acidic, they get fixed in the form of aluminum phosphate or iron phosphate. When the pH of the soil is not correct, then they get fixed in the form of calcium phosphate. So, here what is we can say that when this phosphorus solubilizing bacteria is there in the soil, they solubilize the phosphorus into their available form, and this available form then they become available to the plants. So, when PSB is used with rock phosphate, it has the ability to replace at least 50% of the crop requirement of the phosphatic fertilizers. So, we can see that this phosphatic fertilizer is this phosphatic biofertilizer is very important, and the important phosphatic solubilizing biofertilizers are Bacillus megaterium, Bacillus polymixa, Bacillus subtilis, Pseudomonas strains, Pseudomonas striata, Agrobacterium and Acetobacter diazotrophicus. Other than this, some fungi have also been found to be involved with the phosphorus solubilization, and these are Aspergillus avamori, Penicillium digitalium, yeast and Trichoderma. In actinomycetes, Streptomyces and Nocardia are also one of the important groups which help in solubilization of the phosphorus. Now, why PSB is important? This is very important to discuss, that why phosphorus solubilizing bacteria are so important, because you know the acidic soils that are predominant or found in the tropical and subtropical soils, we see they are extremely phosphorus deficient, and there is like cases of high phosphorus fixation rate in these kind of soils. So basically it is not that phosphorus is not there, phosphorus is there in those kind of soils, but they are not available in the available form, like H2PO4 minus and H2PO4 2 minus form. These are the forms in which this phosphorus is absorbed by the plants. So what happens when these like acidic soils are there, when their pH is below 7, much below 7, then iron and ammonium which are present in this soil, they convert the available form of phosphorus into ferrous or ferric phosphate and aluminum phosphate. So these now when they are in this form, aluminum phosphate and iron phosphate, then plant cannot absorb this phosphate from the soil. Similarly, if the soil is calcareous or the pH of the soil is high in this kind of soil, then also phosphate, whatever phosphate you add in your field in the form of phosphatic fertilizer, then they become calcium phosphate. So here role of this PSB, phosphate solubilizing bacteria, is very important, because they help in solubilizing the insoluble phosphate into the soluble form. Now the mycorrhiza. Mycorrhiza is the symbiotic association between the fungi with the root of the higher plants. So we can say that the 90% of the higher plants have symbiotic association with the mycorrhiza, and mycorrhiza are they are very important for the like in the orchards or in the higher plants, where they not only provide not only they have they have ability to provide nutrients and water, but also they help the plant in developing systemic resistance among them. So this is the mutual relation between the higher plants and the fungi. So here what happens, fungus receives carbohydrates and other growth factors from the plant, and at the same time fungus takes over the role of plant's root hairs and acts as an extension of the root system, and they help in better absorption of the water and minerals from the soil. So we can say that they help in improving the availability of water, availability of nutrient, or we can say water to the plants. So mycorrhiza are commonly divided into two groups, that is ectomycorrhiza and endomycorrhiza. So ectomycorrhiza are those mycorrhiza which are extracellular, which are not present inside the cell, and this endomycorrhiza or those mycorrhiza which are present in the inside the cell. So among intracellular, or we can say among this endomycorrhiza, arbuscular mycorrhiza or AM is the most important one, and they it has been found that they have vesicles in them which help in vesicles and arbuscles are there in them, which help in or we can say which store the different type of nutrients, especially phosphorus, and when the plant has deficiency of these particular nutrients, these arbuscles and vesicles, they make them available to the mycorrhiza, make this phosphorus available to the plants. Now what is the mode of action of the mycorrhiza? So we can say that the mycorrhiza are able to take up or accumulate and transfer large amount of nutrients, that is phosphorus, to the plants by releasing the nutrients in the root cells through their arbuscles. So phosphate transport process in mycorrhiza, it consists of uptake of phosphate from the soil solution with the help of external mycelium, and then translocation of these polyphosphate granules to the internal mycelium, then transfer from the fungus to the host cells occurs. So the role of mycorrhiza in higher plants is that they enhances the uptake of phosphorus, micronutrients and water in the plant. Other than this, they increases the growth of the plant, they enhances the resistance resistance in plants against the root diseases, drought, stress, and they improves the transplanted stock and helps in their better development in the new field. Now, other important group of the biofertilizers are potassic fertilizers. So, we know that potassium is an essential major plant nutrient required by the plants, and potassium solubilizing microbes help to solubilize the unavailable form of potassium to the available form. And as we know that the potassium is absorbed by the plant in the form of K plus. Okay, and muriate of potash is the most important fertilizer, potassium containing fertilizer, which is widely applied in the field. So, here also there is a problem of fixation of the potassium in the soil. So, the role of potassic biofertilizers are very important, because whatever potassium get fixed in the soil, they solubilize this potassium and then they become available to the plants. So, examples of potassic biofertilizers are Bacillus muciginanus, Fretiuria aurantica and Trichoderma herizinum. Now, other group of important group of biofertilizers are plant growth promoting Rhizobacteria. So, plant growth promoting Rhizobacteria are the soil bacteria that colonizes the plant root. They facilitate the plant growth directly or indirectly. They decrease the global dependence of on hazardous agriculture chemicals. They stimulate the plant growth through mobilizing the nutrients in the soil. So, plant growth promoting Rhizobacteria are very important, because they not only make this nitrogen, phosphorus and potassium available to the plants, but also they help the plant in other ways also, like they solubilize the not only they solubilize the phosphorus, they produce the growth hormones like auxin, gibberellin. Also they induce the systemic resistance inside the plants, which help the plant in their proper growth and development in case of like different kind of stresses, like in case of stress, in case of like stress is there due to drought, or in case of stress is there with the salt. So, these are the things which happen, or these are the things which plant growth promoting bacteria help the plant in when the plant is in any kind of hostile environment. Now, the other group of this biofertilizers are organic matter decomposers. Now, organic matter decomposers are those group of bacteria, or we can say microorganisms, which break down the dead plant and animal material and releases the inorganic nutrients. So, this property of the microorganism is very important, because we know that the whatever nutrient is there in the environment, they get locked in the organic form, and plant doesn't absorb the organic form of the nutrient, but they absorb the mineralized form of the nutrient. So, the role of microorganism here is very important, because they help in locking and unlocking of the nutrient, and they help in nutrient cycling, the completion of the various nutrient cycling like nitrogen cycle, phosphorus cycle, potassium cycle, phosphorus, whatever cycle, nutrient cycle is there, microorganisms have very important role to play. So, organic matter decomposers are very important, because whatever nutrients are locked inside the dead organic matter, they help in decomposition of this organic matter. They convert them into simpler forms, and then whatever nutrients are there, these are absorbed by the plants through their roots. So, one of the classical example of this decomposer is Pusa Decomposer, that is a mixture of 7 fungi that produces enzymes to digest cellulose, lignin and pectin in paddy straw. And as we saw that in the winter, the burning of the paddy straw is very common in regions like UP, Punjab and Haryana, when farmers want to prepare their field for the next crop. So, in the hurry, what they do, they start burning the rice straw inside their field itself. So, when they do this, what happens, a lot of pollution is caused due to burning of the paddy straw in the field itself, and lot of organic matter is also lost. So, Pusa Decomposer have been very helpful. So, when we apply this, they help in better decomposition of the paddy straw. So, benefits of these are that they improve the fertility and productivity of the soil. As the soluble, they help in conversion of this organic matter into the manure and compost, which are further utilized in the field, and they help in improving the organic matter of the soil, and at the same time the consumption of the fertilizer decreases after application of this compost, which are developed by applying these decomposers. Similarly, it is an efficient and effective, cheaper and doable practical technique to stop the stubble burning in the field. Similarly, it is an eco-friendly and environmentally useful technology. Now, what are the various types of biofertilizer formulations? So, as we know that the biofertilizers are living organisms which are when applied to the seed, plant or like any kind of soil. So, they help in better utilization of nutrients, and at the same time they also help in absorption of nutrient from the soil, and also they help in better development of the plant. So, I will take a short break. After that we can start again.
जीवन ये प्रकाशित करने राहों को आलोकित करने, जीवन ये प्रकाशित करने राहों को आलोकित करने, हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने, अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने, जीवन ये प्रकाशित करने राहों को आलोकित करने। धन साधन समय की सीमा से विद्यालय जो मुक्त हो, धन साधन समय की सीमा से विद्यालय जो मुक्त हो, कभी पढ़े हम कहीं पढ़े वे विषय की लोच से युक्त हो, कभी पढ़े हम कहीं पढ़े वे विषय की लोच से युक्त हो, मित्रों हम उठे और जागे, मंजिल आने की ध्वनि सुने, हम अपना जीवन स्वयं बने, हम अपना रास्ता स्वयं चुने, जीवन ये प्रकाशित करने राहों को आलोकित करने। राष्ट्रीय मुक्त विद्यालय का है द्वार खुला सबकी खातिर, शिक्षा परीक्षा का अवसर एक बार ही क्यों मिलता फिर, शिक्षा परीक्षा का अवसर एक बार ही क्यों मिलता फिर दुनिया में इसके केंद्र बात, कोई मन चाहा केंद्र चुने, हम अपना जीवन स्वयं बने, हम अपना रास्ता स्वयं चुने, कोई मन चाहा केंद्र चुने, स्वयं बने,
Welcome back. So now we will continue with our topic biofertilizers. So what we till now what we discussed that different kind of biofertilizers and how they are used. So now what are the biofertilizer formulations? So as I said that the biofertilizers are living organisms, but they need certain formulations in which they grow and multiply, and that thing only these formulations only we apply in the soil or in the seed or in the seedling. So formulations is very important. So formulations are further they are of different kinds. So carrier based bioformulation is there, liquid based bioformulations is there, and one other group of biofertilizer we didn't talk earlier, that is multi-strain biofertilizers. So sorry, let me, okay. So a carrier is the second most important ingredient after the living organism or living microorganism, which is used as a biofertilizer formulation after the microbial inoculum. It carries living organisms for a significant period of the time depending on the shelf life and provide necessary support for microbial growth. It is also these carriers are the they are the good carriers, and they can support 1 billion, that is they should support 1 billion, that is 10 to the power 9 cells per gram when the inoculant is fresh and maintained at 100 million cells per gram after production, which is their usual shelf life. The common carriers used for biofertilizers are lignite, charcoal, vermiculite, and this charcoal already I said. Excuse me, actually there is some problem in, so we will continue. Okay. So what are the various characteristics of the good carrier? So it should have high water holding capacity, it should be easy to process, it should be non-toxic to the microorganism, it should be easy to sterilize, and by autoclaving or gamma radiation we should be able to autoclave, or we can say sterilize the carrier. That is very important. It should be available in adequate amount and at a reasonable rate, that is also important. It should provide good adhesion to the seed being treated. It should also have high buffering capacity and high cation and anion exchange capacity. So as per BIS specifications, a biofertilizer carrier should be in powder form and capable of passing through 150 to 212 micron 50 at the sheave. So these things are important, and as I said that the liquid based biofertilizers are also important and they are very much in demand. So liquid biofertilizers contain specific microorganisms, they are very much in demand and popular. The first biofertilizer, that is Rhizobium, produced in 1896 was in the liquid form only and was packed in the glass bottle. So advantages of using liquid formulation is that they have high cell count, they have longer shelf life, they are contamination is not as much as in the case of the solid formulation. They are convenient in packing, and storage and transport is easier for the liquid based biofertilizers. Now I wanted to talk about this multi-strain biofertilizers. Actually these multi-strain biofertilizers are very much in demand these days, because when we use these multi-strain biofertilizers, they don't contain only one strain of the bacteria, there are multi-strain of the bacteria, and each strain has different function. It means if we have applied this nitrogen fixing bacteria, then the other bacteria will be phosphorus solubilizing bacteria. So these things are important. So in multi-strain biofertilizers, we can apply different kind of strains which will have complementary effect to each other, and they can perform different kind of function in the single biofertilizers. So development of products including combined strain, including nitrogen fixer, phosphorus solubilizer, K solubilizer, and growth promoter are receiving interest of various research organizations. These things are very much in demand. So this multi-strain biofertilizers, and where are Merits, there are various merits and demerits of single strain and multi-strain biofertilizers. Like advantages of this single strain biofertilizer is that they are effective strain expected to give the good result, no problem of dominance by the other strain, no problem of antagonism among the strains, no problem of strain mixing, no problem of differential death rate, and less costly. These are the advantages of the single strain biofertilizers. Now, what are the advantages of multi-strain biofertilizers? The advantages of multi-strain biofertilizers are application of consortium and microbes. If one strain fails, then what can happen? Other strain can function in that case. Suppose nitrogen fixer has failed, then your phosphorus solubilizing bacteria can work in that particular environment. No need for separate application of different kind of biofertilizers in this. So, these are the benefits, or we can say these are the advantages. Now, what are the disadvantages? If the strain is non-effective in the case of single strain biofertilizer, then it has the risk of the failure. Now, disadvantage is risk of dominance by one strain in broth culture and creating imbalance. That is the problem with the this multi-strain. This is the common problem in the multi-strain biofertilizers. Now, biofertilizer application technology and practical recommendations. So, before applying the biofertilizer, some recommendation is there, and we should always follow some guidelines before application of this biofertilizers, because as we know that biofertilizers are living organism, we have to treat them as a living being. We cannot treat them as a non-living thing like we treat the chemical fertilizers. So, selection of right kind of biofertilizer is very important. You have to check that you want biofertilizer for what purpose. Other, crop for which a product meant to be used is generally printed on the packet. That has to be you have to check that whatever crop in which you want to use the biofertilizer, that you have to check that whether the specifications are there that this biofertilizers could be used for that crop or not. So, especially in case of Rhizobium, like in Rhizobium, this is meant for use only leguminous crops such as pulses, groundnut, soybean, leguminous vegetables, beans, peas and fenugreek, and legume fodders like berseem and lucerne. So, specific strains of Rhizobium is needed for specific crops according to the cross inoculation group. So, expiry date must be checked in case of biofertilizers. Now, biofertilizer application in Agitobacter and Ajospirillum, there are certain recommendation for before their application. So, these can be used for almost all crops except the lowland rice, where stagnation of the rice is a problem, and also in leguminous crop. In cereals, millets, they can be used in cereals, millets, oilseeds, cotton, and non-leguminous crop like forages and vegetables. Though Acetobacter, they are very can be used in sugarcane. They are we can say that they are somehow specific for the sugarcane. Now, Ajola and blue green algae, they are used, they could be used in lowland rice. Other than this, packet chosen should be should not be bloated, because it shows that some infection is there in your biofertilizer packet. Other than this, the packet contains, in case of carrier based product, should be in the form of moist powder, and powder should be of dry flakes in case of blue green algae. In case of this liquid biofertilizer, the product giving any foul smell should be discarded at once. So, these are the things which we should follow before application of any kind of biofertilizer, and this is the what are the method of biofertilizer application. This is also very important, because application, how do we apply the biofertilizer, is also very important. So, one of the most common technique is the seed treatment, because seed treatment is the best treatment for application and the process is economic. Seed treatment is preferable as the initial population on seed surface will multiply in the rhizosphere, and after 7 to 10 days of the growth, they help in proper fixation of nitrogen in the rhizosphere, thus lowering the carbon nitrogen ratio and thus facilitating the availability of the plant directly. So, what are the steps involved during the seed treatment? So, first we prepare the paste or slurry, mixing the 200 gram of biofertilizer with 300 to 400 milliliter of water. Now this is we can say that 10% solution of jaggery or 5% solution of sugar solution can be applied in particular this as a adhesive in this particular biofertilizer. Then pouring the biofertilizer paste in 10 to 15 kg seeds kept on the gutty bag on the floor and mixing the paste with the seed thoroughly by hand. When drying the treated seed under the shade for 30 minutes, that is very important because that will help in adhesion of your biofertilizer on the seed. Then we sow the treated seed immediately. Now what are the precautions which should be taken during seed treatment is that we have to avoid any kind of damage done to the seed coat. Seed should not be dried directly under the sunlight. Chemical fertilizers and biofertilizers should never be used together. This should be the rule. Now second important technique is soil applications. So soil application of biofertilizer is mainly done for the phosphatic fertilizer, or we can say phosphate solubilizing biofertilizers, and the rate of application varies from crop to crop. So here what we do, 5 to 7 kg of biofertilizer is mixed with 100 to 50 kg of soil or compost for 1 acre of land, and then they are broadcasted, or they can be mixed with the soil. So or they can be used during the irrigation in the standing crop as well. So the other method is seedling treatment method, or we can say that the cutting treatment method, in which what we do, we prepare the culture with in 50 to 60 liter of water, then we dip the set, or we can say the seedling, in the suspension of the biofertilizers for 15 to 20 minutes, and when we dry this inoculated set under shade and then plant the treated sets immediately. So these are the techniques which we apply for the application of biofertilizers in our crop. Another important is seedling treatment, that is similar to the cutting treatment, in which inoculation is done directly to the roots before transplanting the seedling in the main field, and they are recommended for crops like transplanted rice, tomato, tobacco, mulberry, chili, onion, cabbage and cauliflower, and they are recommended for flowers and ornamental plants like roses, jasmine, chrysanthemum, marigold and dahlia. So here what we do, that preparation of culture suspension using 1 to 2 kg biofertilizers into 10 to 15 liter of water is done. Then we dip these roots or seedlings in the suspension for 20 to 30 minutes. After that we transplant the treated seedlings immediately in the field. So these are the basically four methods of application of biofertilizer to your crop. Now how do we apply the Ajola? Ajola is like little bit different than other biofertilizer because they require lot of water for their growth and development, and the Ajola biofertilizer may be applied in two ways for the wetland paddy. In the first method, what we can do, fresh Ajola biomass is inoculated in the paddy field before transplanting and then incorporated as a green manure. This method requires huge quantity of fresh Ajola. In the other method, Ajola may be inoculated after transplanting rice and grown as a dual culture with rice and incorporated subsequently in the field. So these are the two methods which are used for Ajola treatment in the rice field or in some other crop which has water in their field. Now we will do SWOT analysis of biofertilizer. So SWOT is the strength, weaknesses, opportunities and threats. So biofertilizer should be used, but this is not very much common, this is not very much popular among the farmers. So we have to think over what are the strength, what are the weaknesses, what are the opportunities and what are the threats involved with the application of the biofertilizer use. So strengths are the biofertilizers represent a cost effective, renewable and environment friendly crop nutrition input. They provide otherwise inaccessible nitrogen, augment phosphorus for the plant by increasing the solubility of the sparingly soluble form of P, and also addition of, they also helps in secretion of certain hormones which help in better plant growth development. Other than this, as compared to other agri inputs, their application rates and per unit area is much lower, or we can say their cost of application is much lower, being 40 to 100 kg is their application rate per hectare. Other than this, production cost is also low, and it is found that the production cost is around 22 to 25 rupees per kg. It also requires very less space for their storage and for their transportation, this is not a problem. So, these are the strength of the biofertilizers application. Now, what are the weaknesses? Weaknesses are a biofertilizer contains living organisms which can be destroyed under unsuitable storage conditions. As I said, they are the living organisms, so they require proper caring. So, if they are not stored at proper temperature or proper humidity or proper environmental condition, they will be destroyed. Similarly, the shelf life of biofertilizer is low actually, so that's why we have to check what is the expiry period of the biofertilizer, because they have the limited lifespan. Many farmers are not encouraged to use because their impact is not as dramatic as the chemical fertilizers. So, this is the problem, because they are slow growing. So, it is not like the urea or the single superphosphate or muriate of potash that you apply in the field and your nutrient is going to become available to the plant. Actually, it takes some time because it is a biological process. It does all processes of nutrient availability through the biological means. So, it takes time. So, so one has to be patient when we apply the biofertilizer to see the result. Similarly, dissemination and popularization of biofertilizers through extension and mass media is not adequate. So, we have to popularize them. Other than this, the product quality is difficult to assess by the visual examination, and standard products are not very much available in the market. These are the problems. So, and other than this, special training is required at the farm level, and we need to teach the farmers how to apply the biofertilizers in various crops. Other than this, what are the various kind of opportunities? So, huge opportunities lies in the promotion, marketing and application of biofertilizer in India. Since cultivated area in India is very large, and net cropped area of India is 142 million hectare, and gross cropped area is we can say around 190 million hectare. So, we see the capacity or the kind of market in India, the market for biofertilizer is available. The area under seed legumes is around 38 million hectare, which offers a potential market for Rhizobium. Sugarcane also in Acetobacter, as I said, Acetobacter we applies in the sugarcane. So, a large area around 4 million hectare area is available for sugarcane. So, similarly in this area we can promote the Acetobacter. Similarly, in Indian condition we know there is huge deficiency of this phosphorus is seen in all kind of Indian soils. So, use of this phosphorus solubilizing bacteria is very important in these lands. So, we can popularize the use of this phosphorus solubilizing bacteria in most of the Indian soils. Other than this, biofertilizers could become integral part of integrated nutrient management, as Indian agriculture in general and organic farming is in particular is very much in demand in these days. At an application rate of 1 kg per hectare, the current production of around 20,000 tons of biofertilizer is sufficient to treat only 10% of the gross cultivated area. So, we can see that how much opportunity is there in front of biofertilizer production, marketing and their use. So, we only 10% of the area, gross cultivated area is under use of the biofertilizers, and 90% is still unexplored. So, we can think of promoting biofertilizers in these areas. Now, what are the threats? So, after this strength, weaknesses, opportunities, there are certain threats which are lies with the use of biofertilizers. So, we can say that the threat lies in the fact that if proper and timely know-how and quality products are not provided to the farmers, then this environment friendly low cost input will not impact as it is capable of making. So, what happens when we apply these chemical fertilizers and biofertilizers, we start comparison. We compare the chemical fertilizers, we see the instant effect of chemical fertilizers in our crop, but at the same time this application of biofertilizers, after application of this biofertilizer result is slow, we get result after 1-2 years. So, we have to be patient, and at the same time we have to see that the quality of the product should always be maintained at all level. Now, unrealistic high expectations from the biofertilizers, both in terms of meeting the total nutrient demand or ensuring balanced nutrition by using them alone, should also be discouraged. So, these are the threats which are involved with the biofertilizers. Now, I would like to conclude our topic of biofertilizers with following points, that biofertilizers being essential component of organic farming or natural farming, we can say, play a vital role in maintaining the long term soil fertility and sustainability by fixing atmospheric nitrogen, mobilizing the macro and micronutrients in the soil into the available form to the plants. And in context of both cost and environmental impact of chemical fertilizers, excessive reliance on chemical fertilizers is not practical in the long run because of the cost and side effect of these. So, here we have like a huge opportunity to use the biofertilizers in place of chemical fertilizers. I am not saying that we can completely replace the use of chemical fertilizers, but definitely use of biofertilizers will reduce the use of chemical fertilizers. Other than this, with proper advancement in technology and return of awareness, creation of awareness among farmers, application of biofertilizers could be promoted to the larger area. As I said, only in the 10% of the cropped area only this biofertilizers has reached, and 90% of the area is still unexplored, in which we can use biofertilizers. So, with this I would like to conclude and thank you all for your patient hearing. Thank you so much.